So this is a protein. This is the protein melanin. So eye color is determined by the amount of pigment, so the amount of melanin, and its distribution in the iris. The iris is got layers, so it could be like out in front, it could be behind, and so sometimes the light coming and reflecting off of that will give us like a green color. So we're just going to look at specifically blue and brown. So we're going to kind of ignore green because green is due to the distribution of the color in the iris rather than the presence or absence of melanin, okay? So if we look at the alleles for these, H is a normal switch, and little h is a defective switch. Okay. O codes for melanin. The little o codes for no melanin. Okay, so we actually could look at the combination of these genes to determine our phenotype. So this might be something that I would actually have you do on the midterm. So if I had this combination, what would be the eye color? You know, if you have no melanin, your eye's color would be blue, right? So this would be blue eyes. Right? If I had this combination, right, what would that be? Brown eyes. Okay. What about this combination? Oops, sorry. We'll do that one too. That one would be? Well, we're just doing blue and brown, so we're ignoring green right now because it's a different gene that regulates the distribution of melanin. So if it's a defective switch, but they're supposed to produce melanin, but they don't, those genes are turned off, that would be blue. Okay, so those would be blue eyes. Okay. So what is the combination of alleles called? Is that their genotype or the phenotype? Genotype. So I would write this is my genotype. Right? And then the expression of a combination of alleles gives me my phenotype. So my phenotype would be either blue eyes or brown eyes. Okay. So when we look at the evolution of blue eyes, it was believed to be that like up until like 10,000 years ago, there were no blue-eyed humans on the planet. So everybody had brown eyes. And so what they think is this mutation, so this would be a change in the gene, this mutation arose about, it's actually 7,000 years ago. Okay. And so before that time, everybody was brown eyes. Okay. So this mutation arose. And it then got passed down and actually became very prevalent in certain populations. And so if we look at some headlines that you might see about the evolution of blue eyes. The first one is very interesting. Okay. So it is thought to be, this is from like last year, I think. Or no, this is actually later than last year. This was actually big news a few years ago. So they have a single common ancestor. So what that means is all the people that have blue eyes on the planet can be traced back to this person that got that one mutation in that H gene. Okay? So if it arose in a single person, how could it be that if it was a single mutation, how could it be that we could get two copies of the mutation coming together in one individual? Yes, but if that mutation only rose once, what must those carriers be? What was that last genetics problems that we looked at in lab? What? They were related, right? And so we can talk about what is referred to as consanguinity. And if you look at this word, this is kind of a cool word because con means coming together. And what does sanguine mean? Blood. So this is the coming together of bloods. So we don't say that people are inbred, like in the genetics term, you know, like if you're being like a medical person, you can say that they have consanguinity, right? They're coming together of blood. So this is how individuals would come together with a relatively closely related person to have offspring. So if we looked at a pedigree of humans, and let's actually put 2 daughters here. So this would be similar to what we saw in the lab. So those individuals would be first cousins, right? And when they, when you put a double line, that represents that those are more closely related than what you would average see. So this would be first cousins, okay? So let's just say that this mutation arose, and this is just hypothetical, but let's say that this mutation arose here, right? And everybody else was big H, O, O, okay? So we're just looking at the H gene, we're not looking at the O gene. So everybody's gonna be dominant for the O gene, okay? So I could actually just eliminate O altogether, okay? So these 2 individuals, so this is the single mutation. Right? It gets passed down, and so let's say this person has it, and this person has it. And so they inherited it from their father, the same person. And then they passed it on to their offspring. Right? And so only when we get individuals that are genetically related having a child do we see blue eyes arising. Okay, so this would be a blue-eyed individual, okay? But this mutation arose once, okay? Now, if we're not talking about humans, we could talk about what is called inbreeding depression in other organisms, because we might think that this is really gross, right, in humans, but we do this to our animals all the time, right? So we have families of dogs. So we have purebred dogs that are genetically related. They have some of the same genes. And then we breed them together, and then we wonder why they have diseases, right? So inbreeding depression means that the offspring are more likely to have 2 copies of the recessive alleles for a single gene. Right? So generally, if you want a dog that is not going to have to maybe take him to the vet all the time, right, you get a mutt. Because mutts generally are not, they have a lot of genetic variation. So if you get a golden retriever, for example, you might get a dog that is going to get hip dysplasia, which they're going to have problems walking when they're relatively young. And another common mutation that comes together in golden retrievers is juvenile cataracts. So as puppies, they'll develop cataracts and they'll essentially be blind, right? Now you can obviously go to breeders that make sure that they don't have the mutants in their lineage of dogs. So you have to be careful, you know, that's why you get papers that say these dogs have been tested for this mutation, It's never arisen before, right? So they don't have any of these bad mutations, but in general, dogs that are purebred are going to have a lot more health problems than dogs that are not. And that's because of this inbreeding depression. Does that kind of make sense to everybody? Okay. So when we look at this in humans, you know, in the past, we had smaller populations, right? We didn't have this huge global population. You weren't able to choose anybody in the world. Like I could, you know, go to Australia and choose somebody that's so distantly related to me, right? But you're kind of stuck in where you lived and what your community was. And in some instances, people actually chose to keep their bloodlines pure. So there was a misconception in Europe, particularly, where they thought there were royal lineages, right? And so that royal blood was better than non-royal blood. So you had people, this is actually the Middle East or the Midwest, not the Middle East, the Middle East, that's what I want to say. So, but you can see this, so this would be like Egypt, right? So you can see that there's lots of consanguinity, brothers. This is an uncle and a niece, right? You have siblings because they were kind of, they thought that they were better because they came, so they were trying to keep the blood in their family, right? But obviously, then that is going to be very bad. And so actually, most species have mechanisms to prevent inbreeding so that you will actually disperse. So if you think about like squirrels or other types of mammals, they don't, most of the time the offspring have to leave, right? They're actually forced out. They have to disperse to a new environment where they're not going to be interbreeding with their siblings, where they're actually going to find somebody that's genetically different to them to interbreed. So dispersal in a lot of animals helps to prevent interbreeding. So we see these interesting royal lineages producing interesting mutations. We actually see lots of interesting diseases that appear in these families. Okay, before I want to talk about pleiotropy, however, I have another little image here. Oops. Okay. So how can we tell? Oh, look at this. These are the boots I really want. I want those green ones right there. Okay. So Google is so crazy. Anyway, ignore my shopping, this is what I want for Christmas. Okay, so about 7,000 years ago, they actually found a person, and they were able to genetically test them, and they found that using the man's tooth, they were able to get a piece of DNA, and they were actually able to determine his eye color and also his skin color, and most likely his hair color. And so this is very interesting. So if we look at what this person looked like from looking at them, so they're reconstructing it. Okay, so dark skin, blue eyes. Dark hair, blue eyes, okay? So what do you think might have been an advantage to having blue eyes? Why would blue eyes, if it evolved once and started showing up in the population, why might it have become such a popular thing? They're pretty, right? You're like, oh, wow. So imagine like a whole population of brown-eyed men, and then you have this blue eye. You're like, oh, my heart, right? And so they actually think the reason why blue eyes has become so popular, right? So we see it in so many individuals, is because males with blue eyes were more likely to reproduce, and females with blue eyes were more likely to reproduce. So it's what is referred to as sexual selection. There was an advantage, a sexual advantage to having blue eyes. Okay. So they are very startling. I think there was a book that was based on that. Wasn't it called Clan of the Cave Bears or something? Yeah. There was they talked about this blue-eyed person that was so startling to the population. Okay. Okay, so continuing, so that was an example of epistasis. H and O genes kind of interacting to determine eye color. Another example, which is kind of exactly the opposite of genes interacting, is pleiotropy. So that's how you say it. You say pleiotropy. And pleiotropy is where you have a single gene causing many phenotypic effects. So it's a single gene, but it causes a lots of variation, like not just like skin color, but maybe skin color and then like heart disease, right? So you have these genes that can cause lots of differences. And the best example of this in humans is what is called Marfan's syndrome. And it is due to a single defective gene coding for an enzyme called, or excuse me, not an enzyme, a protein called fibrillin. So fibrillin is a protein. And fibrillin is in connective tissue. Right? So any place there's connective tissue in your body, fibrillin is found in there, and then it's like, if it's defective, it's going to cause a defect there, and there, and there, and there, right? So this would be like the characteristics, the phenotypes are long legs and arms. So the bones, right? Another one is heart defect, because there's connective tissue in your heart, and this might cause people to have heart attacks at an early age. Also effects in the eyes. So we have eye lens defect. So they have bad eyesight. So there's an example of a president that we had in the United States that is believed to, after he died, they kind of figured out that he might have had, or that he did, because they looked at it genetically, that he did have Marfan's. And so who can you think of that might have one of our ex-presidents? Lincoln, yes, so Abraham Lincoln. So they did a genetic analysis on him and discovered that he had Marfan's syndrome. So the theory is that he might have probably have died of a massive heart attack, probably by the time he was 50. So he might not have lived that long anyway, but 50 is pretty old actually back then, 1800s. So there's a diagram that looks like this, right? One gene, and so you're like, how could one gene cause all these different changes in your phenotype? And the answer to that is that this is an example of pleiotropy. Okay, next we're going to talk about polygenic inheritance. And so this is where many genes, so think about that word, many genes. Oops, not many, many genes determine a trait. Okay. So a good example of this is skin color. And I'm just going to use a simple model for skin color. It's more complicated than this. There's thought to be at least 6 genes involved. right? So it's not a Mendelian trait because it doesn't follow Mendelian's pattern of inheritance because it's polygenic. Okay. So I'm just going to look at 3 genes. So this is a model, and I'm just going to label them A, B, and C. And I'm going to say that the dominant A, B, C alleles, the dominant alleles, code for melanin. So that's the pigment, okay? The lowercase alleles, A, B, C, code for no melanin. And since C's look so similar, that's why I put the little C over it so I can keep track of that that's a lowercase c rather than an uppercase c. Okay, so something really interesting happens, oops, when we look at this. So you could have individuals that have this combination of, oops, no, sorry, A, B, C, C. Okay. So you can have an individual that has that combination of alleles. They would have a medium skin tone, right? And you could have them mate with a person with the same genotype. This would mean that they are heterozygous for all 3 genes, right? Because they have a big dominant allele and a recessive allele for all 3 genes. Now, the gametes that could be formed, I'm not going to write them all out because it would be a lot because you'd have to distribute those. But I just want to show these 2. So I could have somebody with this combination of gamete. So this one could produce that combination. It could also produce this combination. Why am I doing that? Sorry, it's A, B, C. Could contribute that combination. Okay. And the same thing with this individual. So this individual could have big A. I'm just going to make a Punnett square here. Okay. Does everybody understand why those are gametes? 3 separate genes, right? They only have 1 copy of them. There are no pairs of chromosomes, okay? So watch what happens. So I could have a child that is much darker than either of the 2 parents, right? So that would be a dark child. And you're like, but I thought when you always had an intermediate in color. If you had 2 medium colored individuals, the offspring would always be kind of like a mix, right? That's not the case because of the way we know from Mendel's work that occurs, right? And then what about this one would be like the parent. So it would be, okay. This one would be like the parent. Oops, big C, big C, little c, okay. And this individual would be very, very light-skinned, right? So what that means is these guys could be siblings. You could have a dark-skinned baby and a really light-skinned baby, and they could be siblings, and their parents could be of intermediate skin color, okay? So this happens sometimes with, in this case, it is fraternal twins. Okay, so these are the parents. These babies are not identical twins. They would have to be fraternal twins because they came from 2 different zygotes. And so these are the children. And so they're siblings, okay? But they're dramatically different in color. Okay? So what this tells us about polygenic inheritance is that there is more variation, right? And so instead of discrete variation, we have what is called continuous variation. Okay, so this would be versus discrete. Oops, discrete. Okay. So that is an example of polygenic, right? If I was to like figure out what is the probability of this happening, why doesn't it happen all the time? And that is because it is more rare, right? It's more rare to have a real dark-skinned baby and a real light-skinned baby being produced by these parents, but it can happen by chance, right? So if I would wanted to look at what's the probability of this occurring, I would have to look at my Punnett square, which is demonstrated here as being like little dots. So where there's pigment, that would be the dominant allele. Where there's no pigment, that would be the recessive allele. So these are the two medium colors. And so notice that only 1 out of 64 offspring, because this would be an 8x8 Punnett square, only 1 out of 64 is going to be really light colored right here, right? And only 1 out of 64 is going to be really dark colored. So it's much rarer to have that happen. Most of the time, children, if this is the number of children, the probability of having a child, most of the time the children are of intermediate color, just like their parents, right? But occasionally you get that variation happening. And that's really important because we now know that variation from an evolutionary point of view is really significant. That once it combines in the children, it can be separated back out because of the way that the chromosomes behave, right, during meiosis. The way that they separate back out, okay? So this is really important when we think about racial discrimination, because this is a story of the Roma people in, might have been Germany. So a while back, a few years back, the population in Germany they believed that the Roma people, which are called gypsies, were actually stealing white children. And so they were actually going into these communities and removing children that looked like this. So this child was removed until the people could prove that they were the relatives. So in this case, they're actually the aunt and the uncle, right? They're not the parents. But when we understand the way that skin color is inherited, we would know that this could be a possibility, right? And that it's not evidence of kidnapping. Okay, the reverse was a problem in South Africa when you had, in the 1950s, you sometimes had white people having children that looked black. And so this is a story called Skin. And it's actually was put into a movie, so I think this was on Netflix for a while, and it's called Skin, and it was just talking about the true story of these two white people in South Africa that had a black daughter, or she was more dark-skinned than they were. And so this was during apartheid, and so the black people couldn't do certain things, they couldn't go certain places, they couldn't go to certain schools, right? So they were completely segregated. And so they actually had to put on her birth certificate that she was black, even though they were white, and she couldn't go to the white school. And so they went to court to try to get that changed, and it didn't work. And she actually ended up, in the story, she ended up running away and living with black people. But they couldn't, it was really hard for the society to understand how this could possibly happen, right? But what it must be is that these guys must have had, genes, dominant genes for melanin production that they got passed down, and they had a child that was more dark skinned than they were. So the opposite can happen. So really, skin color is not a good basis for determining populations apart, because there's actually more variation within one population in skin color than genetic variation between the races, right? So skin color is not a good determining factor for race. Okay, any questions about those ideas? Yeah. Yes, it's called Skin. Yeah, it's a really good movie. It was really well acted. And they go to court and they, you hear all the racist ideas about that's horrible. And it caused a tragedy in their particular family. Okay, so let's look at the sex-linked traits. So this is versus autosomal. So what did autosomal mean? Does anybody remember? Not sex-linked, right? So it's on an autosome, not sex-linked. It's not on the X or Y chromosome. So most traits, when we say they're sex-linked, are actually X-linked. And the reason why is that the X chromosome is so much bigger than the Y chromosome that there's more genes there. The other reason why is that genes that are really important are not going to be found on the Y chromosome because females don't have the Y chromosome, right? So if it's important for survival, those genes are not going to be on the Y chromosome because we don't have it. So it's got to be extra genes that then determine the differences between the sexes. Okay, so when we look at X-linked, we can say that a gene is X-linked recessive. Oops, recessive. Okay. Now, a good example of this is a gene that causes hemophilia. So specifically, hemophilia A. And hemophilia means blood-loving. And so this is where the blood does not clot properly and people bleed because of it. Okay. So when it's X-linked, we have to pay attention to whether the individual is male or female because of the differences in the combination of sex chromosomes. So I'm going to write out my alleles here. Okay. So I'm going to put an X and I'm going to put a big H on top of that. Okay. This is normal. I'm then going to put the little h on there, and this is hemophilia. Okay. Then I'm going to draw my genotypes. Right? So that's the combination of chromosomes. So for the female, I could have this combination. I could have this combination, or it could be this combination. So that's all the possible combinations. Okay. So females have 2 X chromosomes, males have an X and a Y. So both of these are going to be phenotypically normal. You're not going to be able to tell the difference. This female is going to have hemophilia. Okay, so these are going to be normal. This is going to have hemophilia. This shows complete dominance. So this individual, we can't tell, but they would be a carrier, right? So they're carrier for the defective allele. Okay. So there's 3 possibilities in the female, but there's only 2 possibilities for the male. So the male could have this combination where he would be normal, or this combination where he has the disease. Okay. So I don't put an H on the Y because this gene is only on the X. So he has only 1 copy of the H gene, not 2. And notice how he cannot be a carrier. So only females can be carriers for the defective gene in this case. Okay. So we can draw a pedigree, and just like we have done previously, we can solve kind of the problem by working our way backwards. So in the past, hemophilia was deadly. So generally, people with hemophilia tended to die. So if I put that X, remember that means that they died of the disease. It is also because males have a 50-50 chance of having the disease, while females have a lower chance of having the disease. It's more common in males, or it's more common in males than females. Okay. So let's look at this. And I always, when I'm doing sex-linked, I always put the Y in, so I just remember. So all the squares are males, so I add my Y. if they have hemophilia, they're colored in, what is their genotype? This one or this one? This one or this one? Yeah, little h. So I'm working backwards, little h. Okay. So I know that this male did not have it, so he had to be big H. Okay. This male did not have it, so he had to be big H. So what must the females be? They are carriers, right? They have to be carriers because the males get their X chromosome from their mother, not from their father. So X, H, X, H. Okay. So now I could say this couple was concerned because they wanted to have another child. So what is the probability if they have another child that this child will have hemophilia? And so we can, in this case, do a Punnett square. So I'm just going to draw the parents up first because this is how I do it even today, right? And because we're using X's, I use my little star for times, and then X, H, Y, okay? So that's the mother and the father. The mother can produce this gamete, and she can produce this gamete, right? So those are the possible eggs. The males can produce a sperm with a big H, right? X with big H or a Y. So this is my Punnett square where I combine my gametes to determine what the possible genotypes of the offspring are. And looks like this. Oh, did I screw that up? I did. Sorry, X little h. That should be an X. Sorry. And then this one's X little h, Y. Okay. Okay. How many on average are going to be male? 50%, right? 2 out of 4 on average. We learned that that's a little bit incorrect when we watched that video about how more male babies are conceived than females. But in general, that's what we would say. And if the baby is male, what's the probability that that baby would have hemophilia? 50%. So if male, if the baby's a male, there's 50% chance that they'll have hemophilia. If the baby is female, what's the percent chance that she will have hemophilia? 0, right? So in this case, we have to distinguish between the sexes, or we could distinguish between the sexes. So if male, right, there's a 2 out of 4, or no, actually, sorry, 1 out of 2. 1 out of 2, or 50% chance of having the disease. If female, there's a 0% chance. If I wasn't concerned about sex, then it would be a 25% chance, right? If I didn't care, if I didn't know the sex of the child, it would be a 25% chance. Of all the offspring that they can produce, there's a 25% chance, and that would have to be male. Okay. So you might have heard color blindness is another example of specifically red-green colored blindness is another example of an X-linked recessive trait. In that particular instance, it doesn't cause people to die, right? They generally don't die of color blindness. And they're just as likely to reproduce. And so then it's more common in the populations, and you could actually get females having color blindness, but they would have to have a father that is colorblind, and they'd have to have a mother who is either colorblind or a carrier. So they would have to have a father that could survive having only one X with a defective allele, and then pass it down. So it's not always that it's only in males. But specifically, if it's a lethal disease like hemophilia, very rare in females. Okay, one more thing.
Can male be a carrier?
No, right? Because he has only one X. No male carriers. In order for a female to get it, a male with hemophilia must be able to reproduce. Because she could get one X from a female, but she has to get the other X from the male. So if a male with hemophilia can reproduce, if he can survive and reproduce, which he can now because we have drugs for it, then it's possible that they would have a female child with it. Yep. They probably would be genetically tested, and you could actually test the sperm, and you could screen babies for that genetic disease to make sure that they didn't have it.
So it's likely that a male would pass on the age and the partner would be a carrier.
Okay, so if she's a carrier, so the question is, if the female's a carrier, right, and the male has it, right, then there's a 50% chance that the daughters would have, this is the cross, there's a 50% chance that the daughters would inherit this allele from their mother and this allele from their father. So then it's equal. Okay, so we know that females have 2 X chromosomes and males have only 1. So what that suggests is that we only need 1 copy of those genes to do work, right? So we have what is called X inactivation. And X inactivation is where we, in females, only in females, 1 X chromosome becomes inactive. And it becomes inactive by being condensed. And so it kind of looks like this. So I'm going to use a color. So if I have 1 X chromosome this color, and if I have another X chromosome this color, this is my female cell, right? You could use X1 and X2 in your notes. So in some cells, we have, so if this is, some cells, it's the X1 that stays active. In some cells, it's the X2. And so I'm going to draw my little X. This would be X1. And this is my X2. And this is my X2. Okay. So what has happened here is that this is blue. It's hard to see, but this is blue, right? And so this is the chromosome that has become inactive. And it condenses down, and it's packaged so tightly that those genes are essentially turned off. And so only the genes that are on this active chromosome are the ones that are expressed, okay? The exact opposite happens here. The blue one, or X2, and then the black one condenses down. Now, when we were talking about, or we watched the video about the Olympic athletes and whether they were male or female, what do you think this is? What is that called? It's a body, so it's called the, not a polar body, it's called a, do you remember? This is what is called the bar body, right? So that is the bar body. So supposedly, if you took one of my cheek cells, if you scraped my cheek cell and you stained for X chromosomes, you would find one darkly staining region in my cell, and that would be evidence that I have 2 chromosomes. So I would hopefully, I might not actually, but I don't know, I think I might. I would pass the bar body test for being female, right? Males do not have bar bodies because they have only 1 X chromosome to begin with, and so that stays active, okay? Okay, so that's the bar body. So let's look at an example of a trait, and this is in calico cats.
So any inactive chromosome like that instead of 2 is called a bar body?
It's the inactive X chromosome. the inactive X. There's no X, inactivation only occurs in the X, not any of the other chromosomes. Only the X chromosome. Yes, so this would also be a bar body. So that's a bar body.
So does it just like shrink like that?
What it does is it wraps itself around proteins and it becomes really tight. So it becomes really tight and condensed, and so no enzymes can get in there to read the DNA. Yep. Okay. So specifically, calico cats that are brown and orange. Okay. So this brown-orange coloration, if they have a white on them, that's another gene, but the brown-orange coloration is X-linked. So I'll write X with a big B is brown. X with the little b, oh, let's put, I think I screwed that up. This should be black. And this should be brown. There we go. Sorry. I realize brown and black are both B's. Okay. So the dominant would be black. What am I doing for brown and black? Brown and orange. Sorry, I'm so confused. Okay, let's scratch that out. This should be, I need to figure out how to use my eraser on here. Okay, so this should be brown, and this should be orange. Okay, there we go. Okay, so if you've ever noticed, there are never calico cats that are exactly the same pattern, right? And calico cats that are brown and orange are always female. So if you see a calico cat that is male and brown and orange, when I was growing up, it was always like we checked. Because the rumor was that if you're a kid and you find a male calico, then you could sell them to a genetics lab for a lot of money, right? So we would go around the neighborhood checking all the calico cats because we were sure we were going to find one that was male, okay? And the reason is this inactivation is random, okay? So inactivation is random. So in skin cells that produce brown fur, it is this X chromosome that is active, and this one's inactive. It's exactly the opposite. Where they have orange, this one is active, and this one is inactive. So it's just due to, during embryonic development, the X chromosomes become randomly inactivated, and then they give rise to the skin, which then gives rise to the pigmented fur, okay? So then we see something that looks like this, okay? And so you do see really cool cats that sometimes, like this one, you have like a line right down their face and it's black and orange. That's kind of rare, right? Because you think if it was random, this one has a little bit of orange over here, right? And then a little bit of black over here. Okay? So that's why that pattern of pigmentation is so unique to each cat. And then that white color is just another gene, right? And this other gene turns off pigment. And so this would actually be genes interacting to determine the difference in calico coloration. So there's something, you know, this could be anywhere in the body. It's just that we see X inactivation, you know, by looking at them, we can see it really clearly in skin color. In humans, instead of fur color, sometimes we have patterns of sweat glands. So in humans, there are patterns of sweat glands. And if you want to know, if you wanted to look this up, it's called ectodermal dysplasia. Okay, so the production of normal sweat glands is on the X chromosome. So if somebody has a mutant copy, most of us have 2 normal copies of it. If we're female, we have 2 normal copies. So let's say X and let's use S. So this is normal. And X with a lowercase. This is a rare mutation. So most of us are this, where sweat glands are evenly distributed over our body, right? But if you are a heterozygote individual, so if you are big X and then little s, right? You could have ectodermal dysplasia, where you could get certain parts of the skin lacking sweat glands. And this, brings to mind the people that have the pigmentation, weird pigmentation, but that's something different. So that is, I don't know if they really truly understand that genetically, but we know that this is X inactivation. So when I was a kid, there was a person on my sister's softball team that when they would sweat, she was a female, when she would sweat, she would like be sweaty on one side of her t-shirt and completely dry on the other, right? So she had sweat glands on one half of her body and no sweat glands on the other. So that is the ectodermal dysplasia.
So do the sweat glands
They would predict that people that have this problem would have a harder time keeping cool. Yeah, so they might not like to run in hot weather, for example, because they might have a harder time cooling off. Okay, so I'm going to stop there for today. And so does anybody have a copy of their review sheet? Because I think I put one thing on there that I didn't go over. I decided not to go over it before the midterm. So just look at some of these. Okay, so there's two things. The last two bullet points on your, actually there's more than two, sorry. Linked genes. We didn't really talk about linked genes, so you can cross that off. Okay, so we didn't talk about linkage. Then we talked about polygenic. We didn't talk about epigenetics, so you can cross off epigenetics. And then we have not talked about the genetics of cancer, so you can cross that off. So linked genes, epigenetics, and genetics of cancer. Okay. So there's a few things that you can so we didn't talk about epigenetics, linked genes, or genetics of cancer. Okay, so I'll see you guys on Monday. Yes.
Okay.
Yep. What is chapter 6? I don't know. I can't remember. Sorry. It's different for every class I teach. Oh, you do? Okay. So it's just through Mendelian Genetics. So 6 months. Oh, it is. Let's say 5 on that. I don't know. I just read there. The next topic. Okay. All right. Yeah. Just through 6.
And I'm having a hard time understanding the I don't either.
So haploid means it has half the genetic material. Diploid means it has twice the genetic material. And the way that it does that is a diploid cell has two copies of every chromosome. A haploid cell has only one copy of every chromosome. So your egg and sperm are haploid. Your skin cells, your liver cells, those are diploid.
Okay, so
So two haploid cells come together to form a diploid cell. We are made up of diploid cells. When we produce egg and sperm, those are haploid.
Oh, okay.
Yeah.
Thank you.
Yeah.
So for the test that we're having, is it over everything? So PowerPoint,
She's so lost where she is. I'm like,
Yep. just that review sheet. We have a review sheet, right?
Yeah.
Those are all the topics, yeah.